as a way of a bit of background before we get started, Aimee joined us. We were just chatting about it when the ESI just opened. So she's very much grown up with her academic career alongside the ESI. And when she started, the labs were quite empty, 2013, and now we're bulging at the seams, and the microbiology group of which she is part has grown and grown over that period. So it's been a fantastic story for us, and wonderful that Aimee's been able to develop her career alongside a growing group, but so successfully. So as she's going to tell us about this shortly, but she's been just, on paper at least, it looks very smooth, moving from one grant to another. So PhD finished, then a NERC Innovation Research Fellowship, and now just got a NERC New Investigator Grant. So it's been doing brilliant work. And she's going to talk about that today. Her title, as you can see, is Not So Natural Selection. Wow, there's loads of people popping up all the time, which is great. Not So Natural Selection, the role of environmental pollution in the rise of AMR. Aimee's going to talk for about up to 45 minutes, and then there'll be plenty of time for questions and comments and so on. You can either put your question in the chat as we're going along, or just stick your hand up and ask your question at the end. So that's enough from me. Welcome to everybody, and I'll hand over to Aimee for her talk.
Thanks, Jane. Can you hear me okay? Good. Okay. Yeah, hi everybody. Thank you for coming to listen to me talk today. So let me just, there we go. Yeah, so Jane's already said a little bit about me, but I did my undergraduate degree at the University of Bath, and then I've been part of the ESI European Centre for Environment and Human Health and University of Exeter since 2013 when I did my PhD. My Industrial Innovation Fellowship finished last year, and then I started my lectureship. So a bit of background, first of all, what is antimicrobial resistance? It's the ability of organisms like bacteria, fungi, viruses, and parasites to survive treatment with antimicrobial drugs. But admittedly, most of the research done so far is on bacteria and antibiotic resistance, and that's what I'll be mostly talking about today as well. In terms of the current and predicted threats of antimicrobial resistance, so there was a paper that came out at the beginning of this year that estimated that in 2019, 1.27 million deaths were directly attributable to bacterial antimicrobial resistance alone, with close to 5 million deaths associated with AMR in the year 2019. And it's been predicted that by the year 2050, this will increase to 10 million deaths per year, making it the leading cause of death worldwide, as well as causing over $100 trillion loss in GDP. So the One Health approach to antimicrobial resistance has really gained traction over the last few years. So this is recognizing that the health of humans, animals, and the environment is all interconnected, and antimicrobial resistance spans across all of these sectors. So it requires interdisciplinary working and collaboration to truly combat the problem. And just to have a quick plug for a continuing professional development course, which is being developed by myself and Anne Leonard, which is going to run in November this year about One Health and AMR. So if you or anyone you know might be interested in this, then please do share. So going back to the title of my talk, what is natural selection? These are just some quotes that I've lifted from the Natural History Museum and National Geographic websites. Basically, it's a mechanism or the engine that drives evolution, so the process through which species adapt to their environments. So, for example, here we have the giraffes with their super long necks that can reach the very top leaves in the trees. And apparently Darwin chose the name natural selection so that it would contrast with artificial selection, which is controlled by humans, and it's sort of selective breeding. So, you know, dog breeds or specific traits in pigeons and that kind of thing. So artificial selection is with humans, but natural selection is supposed to be without any human intervention. And antimicrobial resistance is a natural occurrence. So there was this paper about 10 years ago now that drilled down into ancient permafrost and found that the bacteria that were living there many years ago had resistance to even modern antibiotics. So it's a natural phenomenon. However, it's the human use of antibiotics which seems to be leading to this massive increase in the spread and development of resistance. So in this first figure here on the left, we have when an antibiotic was first introduced into clinic and started to be used widely, so penicillin in 1943, but we actually already knew that penicillin resistance existed before it was employed in the clinic. And you can see since then there have been several new antibiotics introduced and resistance follows soon after. And this second figure here just shows the increase in use of antibiotics in human population over the last 2 decades or so. Generally, it's increasing globally, partially due to slightly more improved access in low to middle income countries. And then finally, antibiotics are also used extensively in animal production. So it's estimated that around 66% of all antibiotics used worldwide are used in for rearing animals, although use in agriculture has reduced in recent years in EU countries due to new legislation. So this figure kind of represents all of the different ways that antimicrobial resistance and antimicrobials can enter the environment. So through these different uses in humans and animals and in plant production, basically the aquatic environment is sort of the final sink for all of these things, including antibiotics themselves. So antibiotics, they're not fully broken down in the human or animal body. And that means that they can reach the aquatic environment either through wastewater treatment plants where they're not fully broken down, or when, for example, animal manure is applied to agricultural land and then you have runoff into nearby waterways. So for a long time, it was thought that antibiotics in the environment were at such low concentrations, they were so heavily diluted that it didn't really matter in terms of whether they could select for resistance or not. But there was this paper that came out again about 10 years ago, which showed experimentally that very low concentrations of antibiotics can select for resistance. So in this figure here, we have the red line, which is the growth rate of the resistant bacteria, which decreases with antibiotic concentration up to the minimum inhibitory concentration for that resistant strain. So the MIC, minimum inhibitory concentration, is the lowest concentration that completely inhibits bacterial growth. And then the blue line is growth rate of susceptible bacteria, which also decreases with antibiotic concentration up to its own MIC. However, they showed that at even lower concentrations than the MIC of the susceptible bacteria, you can get selection for resistance, and they call this the minimal selective concentration. So the way that they did this was to take isogenic strains of bacteria. So it's a single species, 2 different strains which are completely genetically identical, apart from one has a resistance mechanism and the other one doesn't. And then they growed these up at defined ratios, for example, 1:1, in the presence of lots of different antibiotic concentrations, and then looked for a change in the ratio of resistant to susceptible bacteria over time, with an increase in the proportion of resistant bacteria indicating positive selection for resistance. So the way that they sort of summarize this data is to calculate selection coefficients, which is basically just a change in the proportion of resistant bacteria over time, and plot these selection coefficients against the antibiotic concentration with a line of best fit, and where that crosses the x-axis is the minimal selective concentration. So this is just another way to sort of visualize this. This is another paper that came out that same year, but didn't get quite as much attention. But you can see here that they've got a purple susceptible strain of bacteria and yellow resistant, and at 2 nanograms per milliliter of antibiotic, that's where the resistant bacteria have started to outcompete the susceptible bacteria and take over the population. So, as I said, these sorts of papers really raise the question over whether antibiotics in the environment could potentially select for resistance. The problem when trying to relate these kinds of experiments to the environment is that in the environment, bacteria live in complex communities. So are these single species assays representative of evolution that you might see in the environment? So there have been different approaches to try and determine minimal selective concentrations. So some have used published MIC data and tried to extrapolate minimal selective concentrations. There are the competition assays, which I just talked about, and then there are complex community experiments, which is what I've been doing. So the approach that we take is we do liquid microcosm experiments. So we take a complex bacterial community from sewage, so untreated wastewater, inoculate that into a nutrient-rich growth medium and add lots of different antibiotic concentrations, grow that community up for 24 hours. At the end of the 24 hours, transfer a small portion of that community into fresh media and fresh antibiotic, and then repeat that transfer for around 7 days. And at the end of the experiment, you have your evolved community, which you can plate out onto agar plates supplemented with antibiotic to count numbers of resistant colonies. Or you can extract the DNA and use quantitative real-time PCR to count copy numbers of resistance genes and normalize that to copy numbers of housekeeping resistance genes to get a AMR prevalence, or to do sequencing and look at the entire metagenome, looking at changes in resistance genes and community composition. So this was the first paper that we published that was doing this using this approach. So we had our sewage community, and the antibiotic that we were looking at was cefotaxime. The resistance genes that we were looking at were the CTXM resistance genes. And here we've just taken the approach from that original paper and plotted the change in resistance prevalence over time and estimated that the minimal selective concentration was around 0.4 micrograms per litre, which is similar to some concentrations of cefotaxime you can find in the environment, for example, in hospital wastewater. We also found something else interesting in this study. So this figure here is the same data as the previous slide, but it's just the individual data points rather than the average. And it's across the whole range of cefotaxime concentrations that we tested. So again, anything above the line means that positive selection for resistance is occurring. So on average, we saw that even at the lowest concentration of cefotaxime that we tested. But what we also found was that initially there's sort of this nice dose-response relationship where as cefotaxime concentration increases, CTXM gene prevalence increases, but then that sort of plateaued across a very wide concentration range, which is not really what we expected. So we hypothesized that this was maybe to do with the type of resistance mechanism. So the CTXM genes encode for extended spectrum beta-lactamases. These are enzymes which basically break down beta-lactam antibiotics like cefotaxime. So basically, when that degradation happens, you reduce the concentration of cefotaxime across the whole community. So that means that even bacteria that are susceptible and don't produce the enzyme benefit from being in a community with resistant bacteria. And that's one example of why it's important to look at communities of bacteria rather than single species. Another example of that is another study that was done in the ESI, but I wasn't involved in it, was where they did competition assays with isogenic strains, and then they also took those isogenic strains and put them into a complex community and saw if the minimal selective concentration changed. which it did. So when those strains were embedded in the community, the minimal selective concentration increased, again suggesting some sort of protective effect conferred by the community. So that's why it's important to look at these complex communities when trying to extrapolate to the environment. There have been other studies which take other approaches. So like I said, we tend to use liquid microcosm experiments. There have been other studies that have looked at selection in biofilms. So in this study, they used wastewater effluent communities to establish a biofilm and then look at the effect of tetracycline exposure on numbers of resistant bacteria on agar plates. And then they also looked at 2 different tetracycline resistance genes with qPCR. And you can see here at 1 microgram per litre compared to the no antibiotic control at the end of the experiment, they were seeing increases in both of these genes. However, the minimal selective concentration is really trying to capture a change in resistance over time. And with that study, they only looked at the end point of the experiment, rather than looking at the beginning and comparing all of the different things together. So in this study, which was, this paper was led by Isabel Stanton, who used to be with us, but she's now at Centre for Ecology and Hydrology. She did the same experiment, but in our liquid microcosm, exposed to tetracycline, looked at increases in the same tetracycline resistance gene, and when looking at the day 7 data, saw an increase at 1 micrograms per litre, the same as in the previous study. However, when you include the day 0 data, you can see that actually during the course of the experiment, the prevalence of that resistance gene actually decreased massively. So this led us to propose a addition to this now classic figure, which is in all of my talks, and you'll see again before the end of this talk, where below the minimal selective concentration, we suggested that we should also be looking at and thinking about the minimal increased persistence concentration. So what we mean by that is, so selection is where you have enough antibiotics where you see increases in resistance over time, whereas persistence is antimicrobial resistance is still lost, but it's at a slower rate than in the complete absence of antibiotics. And that's important because it means that there's still higher numbers of resistant bacteria than if there were no antibiotics present at all. So what we've been doing is trying to relate all of this to environmental risk assessment. The approach for this is basically where you take a predicted or measured environmental concentration. So predicted, you can try and calculate it from things like the amount that's used, the amount that's removed during metabolism, during wastewater treatment, thinking about dilution in receiving environments and so on, or measured environmental concentrations where you take a sample and then with LCMS or similar, you can actually quantify the amount of antibiotic or whatever other compound is present. And then comparing that to usually experimental data where you've determined where that compound, at which concentration that compound is having an unwanted effect, and then taking the concentration where you know it's not having an effect and comparing that to the environmental concentration. So there's some sort of additional safety factor there. And basically the ratio between the two gives you what's known as a risk quotient. And if that's greater than 1, then that means that there's an unacceptable risk in that particular environment. So the problem with the current environmental risk assessment approach is that it doesn't consider selection for resistance. So on the right here are different tests that have been or are currently recommended for environmental risk assessment of antibiotics. So up until 2018, it was still recommended that toxicity studies in fish were conducted. But in the most recent guidelines, it's recommended that toxicity studies are conducted on Daphnia magna, which is a freshwater invertebrate, green algae, and cyanobacterial species. So only 2 of the recommended tests are on microbes, but the important thing is that none of them directly look at selection for resistance. And so there's a concern that if we were to say set safe release limits or environmental threshold concentrations for antibiotics based on these tests, then you might still be seeing selection for resistance occurring. So a new method has been needed to sort of quickly and cost effectively determine the lowest concentration of an antibiotic that selects for resistance. So the liquid microcosm studies that we do and the biofilm studies and other approaches generally are quite labour intensive. They take quite a long time and they're quite expensive, and usually the kinds of tests that are recommended for environmental risk assessment are quite quick and cheap. So this was what I was interested in trying to do for my NERC Industrial Innovation Fellowship. So again, back to this figure, just to reiterate that the important point for determining the minimal selective concentration is basically where there's a significant reduction in growth of the susceptible strain. And there was also a more recent study where they were trying to model minimal selective concentrations, and they found that a loss in net growth was the most important predictor, important factor for MSC prediction. So taking that and running with it, we developed the SELECT method, which stands for selection in communities of bacteria. So again, we're taking our sewage community, growing it up in nutrient-rich media and supplementing with antibiotics, but rather than doing it in lots of different tubes, which limits the numbers of antibiotics you can test, we put it in a 96-well plate, which allows us to test many more concentrations. And then rather than having to grow up for 24 hours and transfer every day for 7 days, we just incubate that plate in an automated plate reader for around 12 hours and measure the overall growth of that community every hour. And this is what that looks like. So this is the total growth of the complex community over time, and you can see that there's this nice dose-response relationship between antibiotic concentration and growth. So the lower the antibiotic concentration, the more the community is growing. And you can only see this dose-response relationship during the exponential growth phase, was something that we noticed. And what we do is we then take the time point where that dose-response relationship is strongest and then find the lowest antibiotic concentration where growth of the community has been significantly reduced, and sort of term that the minimal selective concentration or effect concentration. And what we did in this paper was we compared that to our more gold standard method. So doing those week-long evolution experiments and using qPCR to look for selection for key antimicrobial resistance gene markers. And we did that for these 8 different antibiotics, which span several different antibiotic classes. And you can see that there's pretty good agreement between the two methods. So generally, we sort of came to the conclusion that a reduction in overall growth was quite a good proxy for selection for antimicrobial resistance. So again, some of the questions that were around this method and the other liquid microcosm experiments that we use is whether these high nutrient and high temperature assays are really representative of what's happening in the environment. And also, would the minimal selective concentration change if we used a different inoculum? So is there an inoculum effect? So we had a look at both of these things in this paper. So for 4 different antibiotics, we tried 2 different types of media. So ISO-SensiTest broth is the growth media that we usually use. Like I said, it's nutrient rich, but it's also designed to prevent binding of antibiotics, so they should be available in the growth media. And we did this at 37 degrees using that media and also 20 degrees. And then we also used a different type of media, which is artificial sewage. And the recipe for which is in one of the currently recommended tests, test guidelines for risk assessment. So it's already an accepted environmentally representative medium that we used, and we did that at 20 degrees as well. And you can see that for the antibiotics that we tested, changing these conditions didn't really have much of an effect on the minimal selective concentration. And then we also had a look at using different inoculars. So we used wastewater influent from a couple of different treatment plants, one in Cornwall and then one in the Midlands, and from a couple of different time points. And we also compared to an effluent community, and again, not really much difference between the different inocular used for the 4 antibiotics that we tested. And then the final thing that we did was to actually use these data to perform an environmental risk assessment. So this is where we were comparing to measured environmental concentrations of these antibiotics. So there's a publicly available database which is created by the German Environment Agency. And we use these data to sort of do a worst case risk assessment. So we used the maximum measured environmental concentration. And then we also did a more reasonable case scenario where we used the median measured environmental concentration. And the numbers on here are the actual risk quotients. So anything greater than 1 is unacceptable risk. And you can see that for several antibiotics, in the worst case scenario, there was unacceptable risk, but also for ciprofloxacin, there was quite high risk even in the reasonable case scenario. So these data were shared with the JRC when they were doing their evidence appraisal for the 3rd watch list for the Water Framework Directive, and our data support their inclusion of ciprofloxacin as a substance of concern. In terms of other impact from this method, we were invited to present this to the All-Party Parliamentary Group on Antibiotics, with a view to creating amendments to the Environment Bill, which was passing through the House of Lords at the time, to try and get recognition of AMR in the environment. The Environment Agency were also in that meeting, and they contacted us afterwards to ask if the method was suitable or if it could be modified to look at selection for antifungal resistance in fungi. So they commissioned us to do a scoping review, and we also have had that published recently as a scientific publication. And then finally, the AMR Industry Alliance, which is the largest international consortium of pharmaceutical companies, asked me to come and present to them and talk with them a couple of times about the SELECT method and thinking around risk assessment of antimicrobial resistance. And that's because they've actually got their own voluntary antibiotic thresholds that they've published for themselves for their antibiotic manufacturing. So there are still quite a few questions remaining for risk assessment. So, you know, there's no consensus on the best method. There are benefits and disadvantages to all of the methods that have been published so far. Something that I didn't really have time to get into in this talk is whether we should be using the minimal selective concentration or a statistically defined effect concentration. So the MSC is kind of, it can be highly influenced by certain things like the number of antibiotic concentrations that you include in the model or how evenly spaced they are. And that can greatly affect the MSC, or you can just statistically determine the lowest concentration that's different to a control. And then also whether risk assessment should be using persistence or selection as the endpoint. So this was something that we discussed in our review paper that came out last year, where we suggested that for sort of classical environmental risk assessment, selection for resistance might be the endpoint that you might want to use. But if you're considering human health risk assessment and environments where humans might be exposed, then it might be more important to look at persistence, because in that case, that's where you're going to have higher numbers of resistant bacteria persisting for longer amounts of time, and therefore increasing chances of exposure. Then there are other questions which are not necessarily to do with antimicrobial resistance itself, but are more general to environmental risk assessment. So Again, this question over how environmentally realistic all of these methods are, not just for determining effect concentrations, but also predicting the concentrations in the environment and so on, and also what the effects of mixtures are. So risk assessment is still focused on looking at the effects of one compound in isolation, whereas obviously in the environment where you have, wastewater effluent going into a river, you don't just have one antibiotic, you'll have several antibiotics, other drugs and other pollutants which might all interact and act differently than they would if they were just on their own. And that leads me on to another point, that antimicrobial resistance is not just to do with selection by antibiotics, although that is the example I've used in this diagram. So there's another evolutionary process called co-selection, where you can basically get increases in resistance to one compound, even if that compound isn't present. So one mechanism is through genetic hitchhiking or cross-resistance. So we have here a bacterial cell with a chromosome in black, and then this extra chromosomal circular piece of DNA called a plasmid. These can be transferred horizontally between even distantly related species of bacteria, but they also often contain multiple different resistance genes, as well as virulence genes and other genes involved in their transfer and maintenance and so on. So in this example, because this plasmid has a resistance gene conferring resistance to antibiotic A and antibiotic B, you only need to expose that bacterium to one of those antibiotics in order for the whole plasmid to be maintained. And so you've co-selected for resistance to that additional antibiotic. Then the second mechanism of co-selection is cross-resistance, where you basically have the same gene, but it has multiple functions. So this here is representing the bacterial cell wall of gram-negative bacterium. And there's a multi-drug efflux pump spanning that cell wall, which can pump out multiple different compounds. So multi-drug efflux pumps are good examples of where you might see cross-resistance to antibiotics or other compounds. So this can work, for example, if you exposed a cell to an aminoglycoside antibiotic, it could co-select for a beta-lactam antibiotic, but it can also occur across compounds. So metals could potentially co-select for antibiotic resistance, and biocides, which are antimicrobials found in household cleaners, disinfectants, detergents, that kind of thing, could also co-select for antibiotic resistance. And there's a question mark over whether there are other types of compounds which could also co-select for antimicrobial resistance. So this is what my PhD students are looking at. So April Hayes is looking at non-antibiotic drugs, so things like your diabetes medication, painkillers, and so on. There's some literature which shows that they reduce the growth of bacteria, and as we know, reduction in growth can be quite important for selection for resistance. So does that mean that resistance is being co-selected? And she's also starting to look at whether there are mixture effects between non-antibiotic drugs and antibiotics in terms of selection for resistance. Then Laura Murray, another PhD student, she's looking at plant protection products. So these include antibiotics. In many parts of the world, they're still sprayed directly onto crops. But also things like pesticides, herbicides and fungicides, so looking at whether they can co-select for antimicrobial resistance. And at the moment, Laura's been looking at comparing the selective potential of antibiotics which are only used in the clinic versus antibiotics which are only used as plant protection products. And then finally, another type of pollution which might have a role in antimicrobial resistance, evolution and dissemination, is microplastics. So Emily's looking at the microbial communities that live on microplastics. These microplastics might also bind pollutants like antibiotics. So whether these are basically create selective environments for resistance. And finally, just a quick slide on the new investigator grant, which was awarded at the beginning of this year. what we're planning to do is to actually start to look at some of these mixture effects using environmentally relevant mixtures, and then starting to look at some more sort of evolutionary questions around whether past exposure history can influence future evolvability. So do communities from chronically polluted environments have different MSCs to completely pristine environments, for example? And also trying to understand what the impact of any potential mitigation strategies might be in the future. So say we decided that concentration of an antibiotic shouldn't exceed a certain threshold in an environment, and we managed to actually achieve that, what happens to resistance? Is it lost over time? Is it maintained? Is it primed then for if there's a future exposure event, and so on? And that grant was supported by the Environment Agency and Roche, and that's with collaborators here at Exeter and also the University of Bath, who will be doing some environmental chemistry with us. So just to summarise my talk today, the environment is polluted with antimicrobials, antimicrobial resistance, and potentially other pollutants, all of which might lead to increases in resistance, and we need to improve environmental risk assessment to try and better understand those risks and mitigate them. And based on the potential risks that antimicrobial resistance is posing now, but also in the future, we shouldn't wait to do that. So I'd just like to thank everybody who's been involved in the work and supported me over the years, as well as the funders. And thank you all for listening, and happy to take any questions.
Great, thanks, Aimee. That was a brilliant talk, very sobering, quite terrifying when you start to think about it, that we don't know all the things that you're trying to find out for us. So fantastic work, lots of claps coming up on screen. And nicely, we've got time for comments, questions, anything anyone wants to add. So please just stick your hand up and put your camera on and ask a question. Or make a comment. No hands are going up. I mean, when you were talking at the end there, I was thinking about polypharmacy, which people talk about now, that people are taking lots of drugs at the same time prescribed by different specialists. No one even looks at what's going on in the human body in terms of the way those drugs interact with each other. And then if you imagine pushing that out into the wider environment, tracking the effects of these chemicals and what they're doing, even in one human body, is tricky, let alone out in the environment. So it's not really a question, but it's a big task. Probably loads more research grants to come.
Yeah, definitely. Yeah, it's massively complex, and I think maybe, you know, that's why it's been so difficult to try and get mixture effects even embedded in normal risk assessment. It's just, yeah.
Yeah. So at least people are starting to think about it now, but then to come up with solutions. And I also wondered about your select method. Is that patented? Is that your baby? Can you patent it? Can you have a spin out? And
yeah, so no, it's not patented. We published it. So the original plan was for it to be, or you know, it still is hopefully the plan, that it could maybe become an OECD test guideline. So for that, they don't really like it to be patented because, you know, it's supposed to be a publicly available thing that everyone can do. So we published it. And yeah, it's freely available, but we do have some ideas about how we could maybe try and use it for different purposes other than risk assessment. So yeah, I think last week at the Microbes in Society IIB, I was having a chat to them about it. So we could see if maybe something else could come from it. But
I was thinking as well, Susanna McClintock is our Royal Society entrepreneur in residence, and she assures me it's not just about patents and private commercial development. There is scope potentially there for sharing that as a public good, as you say. So maybe she would be able to help as well.
Okay.
Sounds invaluable. Anita, over to you.
Hi, just have a quick question. Maybe it will be like stupid or I don't know. So because I'm thinking about like you mentioned about how like antibiotic resistant gene can be transformed with transformation or conjugation. So would it be nice to select to see if these three type of or gene transplant can affect the selection with exposing also to just an antibiotic, like doing the assay with an antibiotic and also with the free DNA in the environment, and see if it does select for different antibiotic, and I mean like with different, obviously, different kind of antibiotic.
So do you mean like having lots of different resistance genes and then you're exposed to one antibiotic and then see if that particular resistance gene's picked up by transformation?
Yeah, because it's obviously it's correlated. So I was thinking about like if you have free DNA in environment and then you select, it can be selected with the one antibiotic or not. Like, I don't know.
Yeah, no, definitely sounds interesting. It would be worthwhile. So yeah, I think there's probably more studies that have looked at increasing conjugation rates rather than transformation, and some of the papers on the non-antibiotic drugs as well, they've shown that they can increase rates of horizontal gene transfer through conjugation. So yeah, definitely.
Okay, thank you.
Steve. Hi, Steve.
Hello, Jane, and hi, Amy again.
Thank
you. Great paper. So much in there. I just wanted to ask a little bit about when you're talking about risk. Is anyone that you know of looking at different kinds of pathways? So I was in Tanzania just before the pandemic, and a lot of antimicrobials, oxytetracyclines going into cattle there, partly because of climate stress. But the kind of microbiologists I was with saying, apart from the drinking of unpasteurized milk, which is the key risk pathway, the rest of it, largely unmetabolized, is going into soils which would fix the tetracycline pretty quickly and neutral, effective. So I was just wondering about those kind of risk quotients. Feel very abstract until you add in the kinds of pathways and so on. And where does that come into those risk assessments?
Yeah, so so you can have risk quotients and do the risk assessment for different environments. So you can do them in surface water, you can do them in seawater, soil, sewage treatment plants, and so on. And some of the things you were saying there about like antibiotics binding to soil and so on, I think is why it's usually preferable to have the measured environmental concentration, although that still doesn't necessarily tell you about the bioavailability in that environment. So yeah, you're right, there's still lots of other issues with the risk assessment as it stands currently.
Well, and presumably when you look at the soil, that's going to vary dramatically. So eDNA work on soil shows the variability, doesn't it? So
yeah,
it would be hard to have a kind of standard measure for something like that. Yeah, hence I can see your test kit, if you kind of had an easy in your pocket model for that kind of testing out in the field, that could be invaluable. Anyone else got any questions, comments? This is the end of term effect, isn't it? Everybody's really quiet. I mean, I suppose I can ask one about the work that you and colleagues are doing on AMR. How many other people out there are doing this kind of work? Is it a very crowded academic field, or is it fast changing, and sort of who's out there doing this work, or are we at the centre of it? I have no feel for that.
So in terms of looking at the more pollution risk assessment side, I think it's pretty much there's us, and then there's another group in Sweden who do a lot of that stuff. But I can see Will's on the call, so I'll just say what he usually says is that we're the largest group in the UK that sort of spans all the different dimensions. So, you know, looking at evolution in the environment, but then also exposure and, you know, trying to come both top down and bottom up to look at it across these One Health sectors. So, yeah.
This is a good place to be doing this kind of work.
Yeah.
And the Microbes and Society network that you just mentioned, obviously that was launched by the university quite recently. Well, I didn't go to the launch, but how many academics have signed up to engage in that network at Exeter? Does anyone know?
Will says it's 500, but he's double counting. But it's quite a lot. It's great. We had a really good meeting last week. Then the Aimee came to, probably about 80 or so people came to that in Exeter. Lots from Penryn, must be said.
It's very exciting to have that concentration of people, and maybe it's happened by chance, but now you've got that momentum to build on it is fantastic. And timely for all the wrong reasons, as you showed at the beginning, and the scale of the problem is only going to get worse. So very proud that you're doing it in our building, but also at our university, so that's fantastic.
Thank you.
Is there anything to share more broadly about the network and the event you had and what's next for it? I'm putting you on the spot here, Aimee, I realise, well beyond what you were asked to talk about.
I'm looking at Steve because he's got his camera on, but Will and Steve are co-leads on it, so they know much better than I do what the future plans and stuff are. Yeah,
so Steve, seeing as your camera is on.
I think we're here to talk about Aimee's work, really, which is really useful. But I think it's really exciting, just because, you know, listening to Aimee and Will over the years as well. You're starting to get a real sense of the social importance of these questions, but also the ways in which we can think really seriously. There's a lot of work on, risk assessments in the social science sphere that's been quite critical over the years of whether these are licenses to pollute. There's more recent work on colonialism and pollution, for example, and how that works within different parts of the world. And so I think there's a real opportunity. It's going to be hard work to bring together this cutting edge science with the kinds of social science, but I think that's one of the points, for me anyway, of the network. I think it's really crucial work. I think it can only kind of make the impact of all the work we just heard about really important. So I guess that would be another question for Aimee, really, about risk assessments and so on. Because every time you speak, I hear this kind of ratcheting up of the kind of issue. Now, I've just got my head around minimal selection concentrations, and now you're telling me about persistence, and I can imagine the kind of world of pollution management and control with their head in their hands, thinking, how far can we go? What have we got to do here? Because every time you run an experiment, the complexity grows in some ways, which can be disabling as well, can't it?
Mm.
Yeah, definitely. So that was something that we were sort of trying to do in that review paper that came out. So we're very much trying to take what was already existing and try and, streamline AMR fitting into it, which was, why we suggested it would be better to use the statistically derived effect concentrations rather than this estimate of the MSC, because, it's all a bit, and it's quite different to what's currently done. And, with the selection and the persistence thing, yeah, I mean, ideally you would just go with the persistence one in every scenario, but is that achievable and is that off-putting when there's currently nothing available? So yeah, it's really difficult.
But it highlights why it's so important that you're part of the conversation around policy that's changing in this field, and so all your partnership work is really, really important to scale this up. Yeah, I can't see any more hands going up, so I think I should probably just thank Aimee again for a brilliant talk. Fantastic work. Lovely that you're doing it all and having an impact with it, because it's so critical. We're going to have another state-of-the-art talk in September when we come back. So our next talk is going to be at the end of September. We're having a 2-month break for summer, but it's been a really great series and lovely to end with your talk for this year, Aimee. Much appreciate it. And long may you continue this great work you're doing. So thank you, and thank you everybody for coming, and see you all soon. Okay.
Yeah. Thanks, everybody.
Bye.
Bye.
Thanks, Aimee. It's a lovely job.